pretend we're reviewing for a test. I'm going to go through kind of the things that I would test you over, and I will be posting an assignment for you to do online tomorrow. So let's start with the structure of chromosomes and a typical chromosome would look something like an X. A little bit of an exaggeration there in the middle. And oops, there is one chromatid, and it is joined to the other chromatid by the centromere. So two sister chromatids joined by a centromere. So this other side of the X is also a chromatid. This is the structure of a chromosome in prophase and metaphase. Two sisters joined by centromere. In anaphase, telophase, a chromosome is one sister. Two sisters separate because the centromere separates. All right, let's look now at why cells need to be small. First thing we said, very important, that diffusion, very efficient at small distance. If things got bigger, diffusion would not get faster. Diffusion takes place at a given rate, and as long as it's a small space, diffusion is a very good way to do it. And that doesn't matter if we're talking about the diffusion of raw materials in and waste out, or if we're talking about the diffusion of information from the nucleus to the rest of the cell, usually in the form of messenger RNAs. So diffusion, very good over small distances, not so much as cells get bigger. The other thing we talked about was the surface area to volume ratio. A ratio is a division problem. So let's look at what happens if we took a cube, which is a simple way to do surface area to volume ratio, three-dimensional cube. We would determine the surface area by the length times the width times 6, because there are 6 sides to a cube. We would define the volume as the length times the width times the height. So if we had a cube that had 1 centimeter size, it would be 1 times 1 times 6 for its surface area, and that would be equal to 1 times 1 is 1 times 6 is 6. The volume would be 1 times 1 times 1, and that would be equal to 1. So if we go over here, surface area to volume ratio 6 over 1, that equals 6. Now let's double the size of that cube. So 2 times 2 times 6 is equal to, let me get my calculator, 2 times 2 times 6 is 24. And then we would have 2 times 2 times 2. Let's see if I can do this one in my head. 2 times 2 is 4. 4 times 2 is 8. So over here, 24 divided by 8, and that is equal to 3, I believe. But let me check. 24 divided by 8, and that is equal to 3, even though I'm not a math guy. Now if we double it one more time, we have 4 times 4 times 6, and that is equal to 96, I believe. 4 times 4 times 6, that is 96. And volume, 4 times 4 times 4, and that is equal to 4 times 4 times 4, 64. So over here, 96 over 64. And if we divide those out, we get 1.5. So, bottom line is, as cells get bigger, surface area to volume gets smaller. And that becomes a problem because there's not enough surface area to provide for the raw materials and the waste generated by the volume of things going on in the cell. New page. Last time too. And let's talk a little bit about stem cells and cancer. Adults. Adults have stem cells. We all have stem cells. All organisms have stem cells. Embryos have stem cells. But what we need to remember is that an adult stem cell, whether it comes from an embryo or an adult, because they both have stem cells, has limited abilities, whereas an embryonic stem cell can become any cell. And that's the real power, we hope, in using embryonic stem cells in the future of medicine. It's important for you to remember that cancer does not only occur in weak organisms. Cancer can occur in any organisms. You just need to accumulate enough mutations in the genes that that control the cell cycle. And speaking of the cell cycle, let's take a look at the details of that. There, that's better. So here we have cells in the process of dividing. These are not in order. First, you should recognize the first cell would be this one, and that cell is in interphase. It has a complete nuclear membrane. The chromosome, the DNA is in the form of chromatin. there is a nucleolus inside this cell. This is a cell that is just going about its regular business. If it was going to divide, it would go next to over here, which would be prophase. And in prophase, that chromatin has gone to chromosomes. The nuclear membrane is still there, but it's dissolving. The centrioles are moving to opposite sides. The spindle fibers are forming. From prophase, the cell we would next find in metaphase. And in metaphase, the centromeres line up on the midline, which means the chromosomes that are still two sister chromatids joined by a centromere are lined up on the midline. The centrioles have migrated to both sides, and the spindle fibers are attaching to all of the chromosomes. So, and then that one would next move over here to anaphase. And in anaphase, we recognize it because of this gap between the chromosomes, and now the chromosomes are one sister chromatid, and they are moving to opposite sides of the cell. And anaphase would be followed very quickly by this one, telophase. In telophase, our nuclear membrane is starting to reform. The chromosomes have gone almost all the way apart, and this structure called the cleavage furrow is formed. So the correct order to put these in would be D, A, C, E, B. This structure labeled X is the centriole, not to be confused with the centromere. The structure Y is again cleavage furrow, and Z refers to chromosomes. Chromosomes, two sister chromatids joined by a centromere in prophase and metaphase, just a sister chromatid in anaphase and telophase. And if a cell is not dividing, it's in interphase. If it is not in interphase, it's some part of division, whether it's prophase, metaphase, anaphase, telophase, or cytokinesis. That cell is actively in the process of dividing. If it is not actively in mitosis or cytokinesis, it is in interphase. Turn the page. Okay, I gotta get a new diagram, so bear with me for a second. Save sketchbook. New. So we've done that. Come over here, and let's get this diagram. To do that, we need to take a screenshot from the selection. Hit copy sketchbook, edit, paste. Actually, let's leave that right there. Get rid of that. Right, so this is a diagram, whoops, of the cell cycle. A in this diagram interphase. So all of this is interphase. The first part of interphase, B, would be G1. C would be S. D would be 2. The region here that does not have its own letter is going to represent mitosis and cytokinesis. So if we go from D, the first part of mitosis, well, we go from D, we'll go in order. E would be the first part of mitosis, prophase. F, the second, metaphase. G, the third, anaphase. And H, the last part, telophase. Now, if we looked at all 4 of those, that would be represented by the letter I, and that is mitosis. And J then cytokinesis. In S, DNA replicated, which means make a copy. In J, cytoplasm splits. A, normal growth. Sister chromatids pulled apart would be anaphase. Got chromatids to the middle, F. Preparation for mitosis, E. Prophase, nucleus and nuclear material divide. Remember, this is division of nucleus.